Gene cloning, introduction of vectors into hosts by conjugation, which is a specific phenomenon that is observed between two bacterial species, maybe the same species or different species, and it has also been observed to be present in non-bacterial cells. And this method or this phenomenon is uh, an example or a phenomenon that falls under what is called as horizontal gene transfer. So let us look at the learning outcomes of this session. So conjugation is a means of horizontal gene transfer with one cell transferring DNA into a recipient bacterial cell through a conjugation tube. And the transfer of this DNA depends on a large membrane associated protein complex. So that is what we understood by conjugation tube. And this complex belongs to the type 4 secretion system, which contains a specialized pilus. Bacteria having F plasmids have TRA genes, which are responsible for the molecular mechanism of conjugation. So any bacteria that have F plasmids are able to carry out conjugation. So these F plasmids basically are conjugative plasmids. Other than transformation, conjugation can also be used for gene mapping. Now, the discovery of conjugation was by Lederberg and Tatum in the year 1946. What they were trying to study was when you take two strains of bacteria and mix them, whether you have an exchange of genetic material between the bacterial cells. That was the thought that was there behind the experiment. So in the experiment, what was taken was a strain called A plus, A minus, and a strain called B minus. Now the strain A minus did not have or were mutated for methionine and bio. So met minus and biotin were mutated, while threonine, leucine, and thymine, the genes were active, were not mutated. In strain B, exactly the opposite was there, which means that the met and the bio were active, whereas the threonine, leucine, and thymine were in a mutated state. Which means that if you look at strain A, the strain A is an auxotroph. Why do you call it auxotroph? Because this strain A will grow in a media that contains methionine and biotin, which means that because it is not able to synthesize methionine and biotin and you provide it from outside, then the strain A will survive in the minimal media supplemented with those genes, supplemented with those products that are not formed by the mutant. The same goes for strain B, that also is an auxotroph. So this strain B will grow in a minimal media which is supplemented with threonine, leucine, and thymine. Which means that if you take individually strain A or you take individually strain B, then they are auxotrophs and they will not grow in a minimal media. If the minimal media are supplemented with the products that these do not form, then they would survive. Now, what was carried out by Lederberg and Tatum was that this auxotrophic strain and this auxotrophic strain were all mixed. And interestingly, what was observed is that on mixing the two strains, some of the strains that were obtained were the wild type strains. That means they were not mutants at all, and they had all these genes in the active form. None of the genes were mutant, which means that what is trying to be understood is that there is a genetic exchange that is happening between A and B to give rise to a wild type. So because the genetic exchange is happening, the wild type has all the five genes being active because These two were obtained from this strain, while these three were obtained from the other strain. And this becomes possible because of a process that is called as recombination. So, exchange of genetic material led to certain bacteria being able to synthesize required molecules for metabolism. And from auxotrophs, due to the genetic exchange, they were converted into prototrophs. So now you did not have any bacteria, or you did not. So the colonies that are growing over here, those colonies are growing on minimal media. You would understand that strain A, which is auxotroph, cannot grow on minimal media. Strain B, which is an auxotroph, cannot grow in minimal media. So what is minimal media? It's just simply salts. But in case of the mixture, what was observed is certain colonies were formed. So when these colonies were picked up and studied, it was found that they had all the five genes active. So it is equivalent to that being a wild type progeny. Therefore, from this experiment, Lederberg and Tatum kind of understood that there can be exchange of DNA between two adjacent or two close by cells. Now, the question that came up next is whether these bacteria are in close contact with each other to enable the transfer of the genetic material from one to the other, or is it that one bacteria releases the the DNA, and that DNA is then, or gene is then taken up by the other bacteria? So to confirm that, another experiment was carried out, and in this experiment what was done is a U-tube was designed specifically to understand whether you need the contact of strain A and strain B to enable formation of the wild type. So strategically what was done is that the U-tube contains two arms, distinct arms, and these two distinct arms are at this position within the two strains is present a fine filter. So the fine filter will have pores of certain size. Now here the selection of the pores size was such that bacteria cannot go from one arm to the another arm. So therefore what was observed is that the fine filter prevented bacteria to move from one arm to another, therefore ensuring that the strain A cannot be in interaction with strain B. So there is no possibility of a contact pos happening between strain A and strain B. So in this scenario where strain A and strain B cannot mix, is there a possibility of recombination? That was the question. So the answer to that was that strain A, which was deficient in methionine and biotin, okay, when this strain A uh, kept in this manner was collected and then plated. Plating was done on minimal media. One could not find a single colony growing. So therefore strain A, which is an auxotroph, will not grow on minimal media. The same was observed with strain B. When it was inoculated on minimal media, no colonies were formed. This is an indication that you have not had a bacteria from strain A go to the arm which has strain B, or vice versa, where strain B has not moved to the arm which contains a strain A. The basic thing, therefore, is that there is no physical contact between strain A bacteria and strain B bacteria in this case. And so, since there is no contact, is there genetic recombination? So, what was observed is that genetic exchange did not happen because of the presence of this fine filter. In other words, therefore, the presence of this barrier does not allow strain A to interact with strain B, which means that contact between strain A and strain B is important for recombination to happen. So the U-tube, which was designed particularly for this function, kind of gave a interpretation of the fact that you need to have the two bacteria in touch or in contact with each other to enable transfer of genes from one bacteria to the other bacteria. So that was something that was concluded by this simple experiment. Now, what is observed is many bacteria have a plasmid that is called as fertility factor. It is also called as F plasmid. Now, interestingly, what is very clearly observed is that all the bacteria that have fertility factor can carry out conjugation, which means that the fertility factor is a conjugative plasmid. So here what is being seen is that the F plasmid, which starts replicating, is able to transfer a copy of that to a recipient cell, which means that a donor cell will always be F plus, that means it has the F plasmid, and the recipient cell will always be F minus strain. So only if a cell does not have the F plasmid will it be able to receive a F plasmid from the donor cell, which means an F plus strain cannot give the F plasmid to an F plus strain. It has to be an F plus strain to an F minus strain. Now, once this F minus strain gets the plasmid, this becomes an F plus strain, and this is now able to be the donor. In earlier times, because you have this as the donor, so this was considered to be the male counterpart, and because this was the recipient, this was considered to be the female counterpart. So therefore this mechanism of bacterial conjugation was considered to be mating between two bacteria, but we know that it is not really mating, it is only just exchange of just the genes from one cell to the other cell. And because this was considered mating, this name fertility factor was used. But the name still holds true for the same. So the F plasmid therefore is a conjugative plasmid. Now, the question that arises is that when the F plus strain donates or gives the DNA, does the F plasmid in entirety move to the F minus? If that was the case, then the F plus strain would become F minus strain. But that is not really what is happening. Uh, F plus strain always retains the plasmid DNA, and it only allows a copy of the plasmid DNA to be transferred to the F minus strain. So this is made possible because of replication through replication, and the replication in most of the bacterial systems that have F plasmid is through the rolling circle, a mechanism of replication. So as we all know that there is an origin of transfer, and at the origin there is a single stranded nick made, and that nick is the point at which you have the conjugation machinery coming and binding, and with the nick one of the strands of the double stranded plasmid is displaced, while the new strand is being formed using this inner template as the inner strand as the it's not inner strand, but one of the strands is used as a template and one of the strands is displaced. Why? Because the new strand that is being formed is displacing the old one, and so therefore this one or this strand that is getting displaced is what will move through the conjugation tube into the F minus strain. So you can very well understand that the plasmid DNA is being retained by the F plus strain, but a copy of that DNA is being transferred to the F minus strain. It goes as a single strand, and when the single strand enters the F minus cell, that single strand is used as a template to synthesize the other complementary strand, and then it circularizes to form the F plasmid. So now you have both having the F plasmid. In natural systems, say for example, many bacteria, including Pseudomonas, have conjugative plasmids. So an oil degrading, say, Pseudomonas species, okay, when the Pseudomonas species are present as a population on oil spills, then it has been observed that the degradative plasmid, which is also conjugative, is transferring a degradative plasmid to all the Pseudomonas that do not have the degradative plasmid, and this enables the entire population of the bacteria, that is the Pseudomonas, to use the oil as a carbon source. So effectively, if this does not have the degradative plasmid, this bacteria will not be able to use the oil. If this has the degradative plasmid, then it will be able to use the oil. But what the Pseudomonas is ensuring is that through conjugation, it passes on the degradative plasmid to all bacteria, so that all bacteria are equipped to use oil as a carbon source. So that is the advantage of conjugation in natural systems. But in gene cloning, conjugation becomes a means of transferring a gene of interest from one bacteria to another bacteria. So the donor cell, as we mentioned, retains the F plasmid while a copy is given to the recipient. Now, how does the entire machinery work? So the F plasmid has genes that gives rise to several proteins that ultimately form what is called as the conjugation tube. So this basically you can see is that several proteins come together, and this one over here is the pilus, which is able to um, you know get push in and pull out. So therefore you can see how the DNA is being pushed out from one bacteria to another bacteria, and this becomes therefore the conjugation tube. So you can very well see how the conjugation tube is being formed, okay, and you have what is called as the secretion system. This secretion system will push out the DNA. So you can see a relaxosome is formed. This relaxosome will bind to the type 4 secretion system, which is basically the motor part of the pilus, and through this conjugation tube you will have the DNA being pushed out. So it will go from one cell to the other cell, but of course retain its copy within the cell. Now let us understand the fact that, and this is something that is happening, that a plasmid can be episomal as well. So it has been observed that F plasmid is episomal. What do we mean by that? It means that the F plasmid can integrate within the host genome, as you can see over here. Now, when this is the case, then the plasmid is called as the F prime plasmid, because it is no longer an independent autonomous system, but it is now integrated within the host genome. So therefore, this form of the plasmid is what is called as F prime plasmid. Now, when time is conducive, or when the systems are conducive, or when the environment is conducive, then the F plasmid can excise itself out from the host genome, okay, and become again an autonomous system. Interestingly, what has been observed, that if using the F prime plasmid, there is a nick and rolling circle mechanism of replication begins, then from one part of the F plasmid, you will have the displacement of one of the strands, and using this entire template, which is the host genome, you would have the replication beginning, which means replication will not only be of the F plasmid, it will also be of the chromosomal DNA. So if that happens, then what can happen during conjugation is that you not only have the F plasmid entering, so as you can see, one half of the F plasmid entering, but you will also have the chromosomal DNA entering the recipient cell. And this will continue till you reach the other half of the plasmid. So this is basically going to lead to transfer of the entire host genome from one cell to another cell. It means what we understand is that in the recipient cell, you have duplication of the entire genome happening. So a copy of the host chromosome with the F integrated, that means the F prime, remains in the donor cell. It will always retain a copy, but another copy has been transferred to the recipient cell. What does this indicate? This indicates that you can have a bacterial gene getting transferred into another bacteria. Now there are also what is called as HFR strains. What does HFR stand for? It is high frequency recombinance. So when you go to this, obviously what you are having of the plasmid and the chromosomal DNA, this entire thing is what is called as the recombinant DNA, and the exchange of genetic material between the two cells have occurred. Now, what is HFR strains? So, when you have the plasmid integrated into the host genome, and every few minutes, if you check, you will find that sequentially or in order, the genes are getting transferred, genes of the host bacteria are getting transferred into a recipient cell. But obviously, it is a copy. One must remember that. So such a kind of study is what is called as interrupted mating experiment, and this leads to formation of several HFR strains. What do we mean by that? Suppose you have the transfer beginning and you interrupt the transfer at the 10th minute. Then, because the mating has been interrupted, you will not have the entire genome being copied and sent to the recipient. A part of the genome will be copied and sent to the recipient. If you let it go on for another 5 minutes, then you will have a longer chromosomal DNA get into the recipient cell. So, as you keep increasing the time period given for transfer of DNA, you will find eventually that one by one the genes of the chromosomes are transferred to the recipient cell. So like for example, in this graph, you can see that closing to 10 minutes, one finds that this gene has been transferred. Then this gene is getting transferred, then the lac operon is getting transferred, and then you have the gal as well getting transferred. So at the 25th minute, you can observe that the gal is also getting transferred. So which means that interrupted mating has shown that in a certain order, you have this entering the recipient cell first, then this enters the recipient cell second, this the third, and this the fourth. Because of this order in which the genes are being transferred from the donor cell to the recipient cell, it is possible to map the chromosomal DNA. So this is an extremely important thing. Now, for example, in the 10th minute, if you only have a copy of half the plasmid and the and this gene, then obviously that is one recombinant. If you have the half the plasmid and these two genes, this is another recombinant. If you have the plasmid and three genes, that is another recombinant. And if you have the plasmid and you have all the four genes present, that is another recombinant. So therefore, it is called as high frequency recombinant. Now, mating, when disrupted at specific intervals, as mentioned, leads to alleles appearing in the recipient sequentially or in a certain order. So, as you can see over here, at 0th minute, you don't have any of the genes coming in. 5th minute and 10, between 5th and 10th, you have this gene coming in. Between 10 and 15, you have this gene coming in. Between 15 and 20 minutes, you have lac coming in. And between 20 to 25 minutes, you have gal coming in. So you can see one after the other, if you allow the recipient to receive the DNA for as long as 25 minutes, you may be able to get all the genes. But if you only allow till 10 minutes, you will get only one gene. If you allow till about 15 minutes, you will get the second gene as well, and so on. So a linkage map of E. coli can be constructed. depending on the sequential or the order in which the genes are being transferred, you can actually say this gene lies of between this gene lies here, this gene lies here. So you can actually map the genes in the DNA present. And so effectively, conjugation has been a means of mapping genes. So just looking at the bacterial conjugation, a summary of it, you can see that you have an F plus donor, okay? And here you can see that you also have an A plus. So when you have the conjugation and transfer happen, the F plasmid goes in, okay? Now, so you have over here, because of no integration of the F plasmid into the host genome, you will have from an F plus the plasmid being transferred to an F minus, and because that happens, you have got both the cells becoming F plus, F plus. But the bacterial DNA is not getting transferred. So what was F plus A plus will remain as F plus A plus, and the recipient will only become F plus A minus. So earlier it was F minus A minus, but with the transfer of the F factor, it becomes F plus and A minus. This is very clearly indicative of the fact that because the F plasmid has not integrated into the host genome, you don't have any component of this bacterial host DNA being transferred to the recipient bacteria. But however, if you have the F plasmid have getting incorporated into the genome at a specific point, okay, and using the F plasmid origin, if the rolling circle mechanism begins and the displaced DNA is transferred to the recipient cell, wherein the single strand is then getting converted into double strand. But in the process of doing so, what has happened is this A plus, which is present in the donor cell, a copy of that is also going into the recipient cell. So in this case, what is going to happen is you have F plus A plus and the recipient is F minus A minus. But now what is going to happen is both the strains are going to become F plus A plus or F minus A minus. So that is something that is interesting to note. Genes transferred are from origin in a specific order. And this is basically explained by a rolling circle mechanism, very clear. If you have the origin of transfer over here, a single stranded nick is formed, then you have a helicase and a DNA polymerase using this strand as a template, synthesizing a new strand, and this strand is getting displaced. Now therefore, part of the plasmid is getting displaced. This is the front part of the F plasmid. But then, because it was integrated within the host genome, you can see how the genes of the host are also getting transferred along with the front part of the F plasmid. If an entire cycle of replication is allowed, then you will have an entire chromosomal genome with the entire plasmid DNA getting transferred to a recipient cell. If it is interrupted, then you will not have the entire copy going in. You will have only parts of the host genome and the plasmid DNA getting into the recipient cell. Therefore, the conclusions are conjugation, a horizontal gene transfer molecular mechanism, enables exchange of genetic material between two bacteria, conferring the ability to form a large protein complex with a specialized pilus having type 4 secretion system are genes that may be present in chromosomal DNA. So therefore, a conjugation can depend on genes present in the chromosomal DNA or as we just observed in plasmids. Several bacteria have F plasmids that are able to replicate their plasmid by rolling circle mechanism, and the copy is transferred to the bacteria that does not have F plasmid. So the F plasmid can be episomal, and because of this, you have HFR strains getting formed in which the F plasmid is integrated into the genomic DNA, and this enables transfer of genes of chromosomal DNA from one bacteria to another bacteria. So conjugative plasmids, therefore, are genetic tools that can be used for transformation with the possibility of stable transformation and gene mapping. Stable because the plasmid can get integrated within the host genome. Thank you.